Hey guys, and welcome back. I'm Matt, and here today in our workshop, I'm going to be working on a project to do PCR. PCR, polymerase chain reaction, is the most powerful tool in biotechnology today. What it involves is replicating a DNA sequence exponentially to make multiple copies for multiple reasons. The way it works is we take a large piece of DNA, even a full genome, design primers that will bind in specific locations, melt the DNA, the primers bind, and we copy the area in between them. After a few rounds, we just end up making multiple copies of that region, and that number will grow exponentially, giving us plenty of DNA to work with. When I call this the most powerful technique in biotechnology, I'm not just saying that. This is used in forensics to get enough DNA for DNA testing. It's used in laboratories to replicate genes for insertions into plasmids. It's branched into several other technologies. Site-directed mutagenesis, a way of changing genes, is literally just PCR with specialized primers. qPCR, or reverse transcriptase PCR, to copy RNA instead of DNA. This is an absolutely wild technology. But the thing that makes it possible is that polymerase. Polymerase being the enzyme that copies DNA. And the polymerase used in PCR has to be able to survive the heat that melts DNA. The man that made that breakthrough went on to get a Nobel Prize and all kinds of things for his work. But the machine that runs the PCR only has to get hot and get cold. It does so successively and quickly, but just doing that, it ends up costing over $1,000. These prices are ridiculous in my mind for a machine that just does something like that. And so I said, I bet I can replicate this technology. I bet I can make this here and make it available to anyone that wants it. So my first attempt was to just use pieces from the 3D printer. You've seen it around the workshop before. It gets hot, it gets cold. I've used it for heating applications as well as for actual 3D printing in the past. I have an Ender 3, so of course I have all my old pieces as I've slowly upgraded to newer and fancier pieces. That's the whole point of running an Ender 3, is to spend all your time upgrading it instead of printing. This heater sink, though, was a bit of a holdup. Getting cold is a bit harder than getting warm, I guess. We heat it up very quickly, but I believe this heat sink is designed to transfer heat away from itself, keeping the cold end cold and the hot end hot. What I need is something that is going to pull heat right out of my heater block. I've got a CPU cooler coming in, ordered on AliExpress, and I will be seeing all of you soon. Alright guys, the CPU cooler is here and I've been working on it for a couple of days because there have been a few weird little hiccups. First off, 4 wires coming out of each fan. I assumed a positive and negative for the LEDs and a positive and negative for the fan. No, that's not the case. It's one that goes to red, or one that goes to black, and 2 that go to red for each fan. And then I guess a data pin that doesn't seem to be necessary for constantly on operation. Won't run without all of that, so we will have both fans and LEDs. And so this is going to be our thermocycler. I don't know. I'm down for it. Also worth noting, there was a third fan when I started. This one arrived completely dead. I'm going to be writing to AliExpress, telling them they owe me a fan. But the simple truth is that we can probably run like this. There was no math or reasoning behind why I got a 3-fan, 6-heat pipe CPU cooler, except cost calculations. No actual idea how much cooling we're gonna need, or if this will even be enough, even if it was fully functional. So let's find out. All right, guys. So here we are watching the first test live. We have the heater going. Time to power up the fans. And this first test will, of course, just be with continuously on fans. Look at that. We're already nearing our peak temp. That got kind of jumpy there for a second. All right. We're still kind of surpassing, but now we're surpassing and dropping below it all within a couple of seconds. All right. The first minute's up, so now we're into that ramp down. Remember, our goal here is to get into the 50s as quickly as possible. And before, that was taking about 5 minutes. Now we're at 90 seconds. Okay, guys. I saw 155 go by. I'm going to have to look at my thermostat and make sure it's doing okay, because some of these numbers it's generating are a little out of order. But the general pattern seems to be that we got down to temperature in about a minute. That is still a lot slower than a commercial thermocycler, but for this, I think that's a success. So this heatsink sure has grown a lot. Walk through the current setup. We have my Arduino running the programming. Running 2 separate MOSFETs for 2 separate power supplies. One controls my heater block and one controls these fans. These fans are directly placed on top of where the PCR will be performed in this little 3D printed block I have down underneath the cooler. And we were able to sort out all that jumping around in temperature. Turns out, since I don't actually build PCs, I didn't understand how to use thermal paste. Go figure, that was important. Other note though, if I added too much thermal paste, this actually cooled it down so much I could not reach the max temp. So there have been some adjustments to the programming along the way, and now we get hot and get cold. I don't think you guys want to just stare at my computer screen for the rest of this demonstration though. So I've got a little bit of a surprise for you all. Alright guys, so that's the thermocycler, but I don't know if you can tell, we are not in the Nodal lab anymore. We have gotten into an actual lab, and we are going to actually be able to test and run this thermocycler against an actual for sale model. This thermocycler, now this does have a lot of features that I am not putting into mine, so when we do our final price comparison at the end, I'll keep that in mind. This does have 3 different compartments, each of which is capable of running different programs. It stores multiple programs within itself, all ready to go, and it is capable of active cooling down to 4 degrees Celsius. So, let's see if those features are actually worth the money and get some actual results on our homemade model. All right, little secret here for forbidden biochemistry techniques. Whenever you're using a pipette, you're supposed to get that little drop and put it into the base. That way you're pipetting into the liquid for most accuracy. But I'm just going to tell you now, when you're pipetting 1 microliter, the difference between 1 and an inaccurate 0.9 is not nearly as great as the difference between 1 and the pipette messed up and you got 0. So it is perfectly acceptable to stick your little droplets to the side like this so you can visualize that you are actually adding at least some of your mixture, as long as you centrifuge them all down to the base before you start your reactions. And in they go. We're off to the races. And our samples are off and running as well. So just a reminder, this is the thermocycler we are competing against. $12,000. I have no clue why the other version is always more when it only has 2 wells, but that's consistent with the prices from multiple places. 19,000, or ours with the triple wells, 12,000. Very interesting way to price things. So one way to know that the PCR was successful is to run an agarose gel. All this is a gel where we put the DNA in the top, and then it is pulled through by an electrical current. DNA has a negative charge, so it will run towards this positive end. And since it's just current pulling it through a gel, it will sort it all by size. We know the size of the target we were trying to amplify, so we'll be able to determine how much we were able to get from the PCR. Alright guys, so back from the lab, and this is the gel we made. There are some issues. As you can see, there we have our ladder. The fragment we were trying to copy is 1,000 base pairs, or 1 kbp. And as you can see, the commercial thermocycler, both samples successfully worked, but our homemade one, they did not. Not losing hope yet. There's an old saying, the difference between practice and theory is greater in practice than in theory. So for a first outing, a failure like this isn't the worst thing to happen. I think we can come back tomorrow and try again. We tried this too many times though, we're eventually going to get told we're wasting reagents and need to come up with something. So let's sit down and make sure we know a way to fix this, and then give it another go. Alright, so we've made some important changes. After that last failure, we've moved the site of the thermistor to one of the capsules. These little PCR tubes were not heating up at the same rate as the block, so it's important that we get our temperature from inside one of the PCR tubes. This does mean that we'll only be able to run 1 sample at a time now with this model, but it's what we've got. The other issue with not heating up at the same rate as the block was that there wasn't thermal transfer between the block and the tubes, and there was a huge delay. Since the block still was getting hot, that was causing some issues. It's a bit improv, but what I've done is I've fixed that by filling the area between the tubes and the block with a whole bunch of thermal paste. Let's see if this goes. Take 2. And that deal with the thermal paste is so jank, but these numbers are right where we want them to be. And the thermistor is now directly inside a tube adjacent to the tube we're working on. So this should do it. So guys, here's the new gel. And look at that. Both lanes this time are from this thermocycler. I didn't waste reagents rerunning on the commercial one. As you can see, it worked great. And today, so did mine. We have these 1,000 base pair strands. Little bands of DNA right here. That means that we're successfully replicating our target piece of the genome with a thermocycler that we built, when I came out and did the math, less than $100. Compared to the thermocycler we competed against, that is insane. Compared to even a basic thermocycler though, we under 1/10 the price, maybe at like 3% of the price. So for those results, I'd say we did it. We successfully showed that PCR can be done with components off the shelf or from around your workshop if you're established enough to have one. Admittedly, being a slightly established workshop did help, but even then, a lot of the stuff I have here came from kits because these are little cheap components. So that $100 price does involve purchasing the Arduino kit, purchasing a full Ender 3 hot end, the way that you would have to do it, or to do it successfully. You could also buy all the little components and get 5 to 100 in batch orders, and I just don't know that you need 5 of these. I don't know if you need 1 of these, but that's the point, is if you do, now you have one. If you have any reason to want to do PCR at home from your own curiosity to the advancement of science, it's now available. Like my other inventions in the past, I'm going to be making this as publicly available as possible. I'm going to try and make a parts list for everyone, but once again, I don't know that I recommend using the exact parts that I did. If someone knows more about thermal transfer than I do, and they want to take on this project, feel free to contact me, or even just show me afterwards what you've done. What I think is going to be really valuable is the code that I've got running this, so I'm going to make sure that that's linked as soon as possible. And guys, until next time, stay hungry.